And so do I hit share screen now? Can you guys all see that? Perfect. Thank you so much, Jay. It's a real pleasure to be here, and I'm really honored to be asked by Alex's Lemonade to speak about our work. I'm also really grateful to the foundation for all the work that they've done over the last 20 years. Amazing that it's 20 years. Supporting the work of myself and my colleagues. It's no exaggeration to say that none of the work that I'm presenting today and the work that I've done over the last 20 years has been heavily supported by Alex's Lemonade Stand Foundation, and they really are doing a fantastic service to the community. So thank you for that. As Jay said, I'm going to talk today about the mechanisms by which pediatric brain tumors, and in particular medulloblastoma, evade the immune system. And then towards the end, some strategies that we can use preclinically, but also clinically to try to overcome this evasion and promote tumor rejection. Let's see if I can advance that. There we go. So there's an old pop psychology quiz that some of you may be familiar with, in which people are asked, if you were able to get a superpower, which would you prefer, flight or invisibility? And the notion is that this is supposed to reveal something deep or fundamental about your personality. If you choose flight, that must mean you're bold and extroverted and heroic. And if you choose invisibility, that must mean you're shy or introverted or even kind of sneaky and underhanded. And so, not surprisingly, when people are given this survey, the majority say that they would prefer flight. But the truth is, invisibility is really undersold. I think there are some significant advantages to this superpower. In particular, flight allows you to go places where you could already go, but just get there faster. Invisibility allows you to go places where you couldn't otherwise go and see things you couldn't otherwise see. And so I think this is a tremendous power that for a superhero or for a supervillain can make the difference between victory and defeat. Now, cancer is the ultimate supervillain, and cancer has many superpowers, but among them is this quest for invisibility. Cancer knows that if it can hide from the body, hide from the immune system, it can do whatever it wants, it can wreak whatever havoc it wants, and there's nothing we can do about it. And so it behooves us to try to understand the mechanisms by which cancer tries to achieve invisibility, and then try to find ways to circumvent this so that we can attack and eliminate the disease. So we've been studying this for several years now in the context of medulloblastoma, which most of you know is the most common malignant brain tumor in kids. This is an extremely aggressive tumor that, if left untreated, will grow rapidly from where it starts in the cerebellum, but it will spread to the forebrain and down the spinal cord, and eventually, if untreated, will kill the patient. Now, we almost never leave children with medulloblastoma untreated. In fact, nowadays, kids who are diagnosed with medulloblastoma receive a very intensive treatment regimen, including surgery, craniospinal radiation, and a cocktail of really potent chemotherapeutic agents. And this combination of therapies certainly prolongs survival in the majority of kids. But many patients relapse and ultimately die of their disease. And those who survive often suffer severe long-term side effects from the treatment, including cognitive deficits, endocrine disorders, and an increased susceptibility to secondary cancers a few years down the road. And so we clearly need more effective and less toxic therapies for this disease. And this has been the goal of my lab for many, many years. Now, I've been talking about medulloblastoma as if it were a single disease, but in truth, through extensive genomic and epigenomic analysis, we now know that medulloblastoma is really probably has at least four major subgroups. These are almost as distinct from one another as tumors in different tissues. There are tumors that are driven by the Wnt signaling pathway, tumors that are driven by mutations in the sonic hedgehog or SHH signaling pathway, a set of tumors that are termed group 3 that are largely driven by overexpression of the c-Myc oncogene, and a set of tumors called group 4 that have drivers that we're only beginning to understand. These tumors not only differ from one another in terms of the molecular aberrations that cause them, they also differ from one another in terms of the prognosis of patients who are diagnosed with them. So for example, patients who have Wnt-driven tumors have a fairly favorable survival. Most of these patients end up living normal lifespans as a result of the extensive therapy they get. And in fact, these patients are now the subjects of trials that are aimed at de-escalating therapy to see if we can allow them to survive without the significant side effects that they suffer. By contrast, patients with group 3 medulloblastoma shown here have a very poor prognosis. These are the patients who relapse the most frequently. They have metastases that go down the spinal cord. And these are the patients who tend to die of their disease. And so for these patients, we definitely don't want to de-escalate therapy. We actually want to intensify or at least find more effective therapies that can cure some of these kids who are dying now. So I'm going to focus today mostly on group 3 medulloblastoma, a little bit on sonic hedgehog-driven medulloblastoma. And so group 3 medulloblastoma represents about 20 to 30% of the human disease. These tumors are often characterized by large anaplastic, that is nasty, bizarre looking cells. The tumors often recur and metastasize much more frequently than other classes of medulloblastoma. And as I mentioned before, they're associated with amplification or overexpression of the MYC oncogene. So a number of years ago, in order to study this disease, we decided to try to develop animal models because those models didn't exist. And so we took the following strategy. We isolated neural stem cells from the cerebellum of neonatal mice. We infected them with viruses carrying oncogenes that we thought were drivers of group 3 medulloblastoma. And then we transplanted these cells into the cerebellum of another animal and monitored the animal to see if they developed tumors. And indeed, if we infected cells with viruses encoding the MYC oncogene and a mutant form of p53 tumor suppressor, these cells would go on to form tumors in 100% of mice. If you infected with either of those genes alone, you wouldn't get a tumor. So this is a model that we refer to as the MP or MYC-p53 model. More recently, we discovered another oncogene that might play an important role in this disease, and that's termed GFI1 or growth factor independent 1. And we found that when we overexpressed MYC and GFI1, this could also drive tumor formation in 100% of mice. And we refer to this as the MG model or MYC-GFI1 model. Now, we've used these models extensively to study various aspects of the disease. We've defined an important role for PI3 kinase signaling in tumor growth. We've done high-throughput drug screening to identify histone deacetylase inhibitors as effective drugs against this disease. And in fact, these cooperate with PI3 kinase inhibitors. And we are in the midst of some intensive studies on the mechanisms of metastasis in these animals as well. But a number of years ago, my postdoc, Alex Garanchet, decided that she wanted to use these models to study immune responses to medulloblastoma, recognizing the important role of immunotherapy in treatment of other cancers. But there was one problem with this model for studying immunotherapy, which I didn't go over before, but that's illustrated here. So when we first generated these models, we would infect stem cells with oncogenic viruses and transplant them into the cerebellum of a NOD-SCID IL-2 receptor gamma knockout or highly immunodeficient host, and then transplant the cells into these mice and monitor them to see if they develop tumors. Well, they all developed tumors, as I showed you before, but we were doing this in a mouse with no immune system, and that kind of model is not really helpful for studying interactions between tumors and the immune system. The reasons we were doing this are not worth going into, but suffice to say it was a lucky coincidence that we put these cells into mice with no immune system and allowed them to grow unmitigated. Because when we tried to grow these tumors in mice with an immune system, we had a bit of a surprise. We found that the MYC-p53 tumors were capable of growing in immunodeficient mice, NSGs, as well as in wild-type immunocompetent mice, the C57 Black 6 strain, here with an albino mutation so we can image them better. In contrast, the MYC-GFI1 tumors, which we knew grew in the immunodeficient mice, could not grow at all in mice with an immune system. So if we had tried to make this model in an immunocompetent mouse, we would have failed and we would have concluded that GFI1 was not an oncogene for these tumors. In any case, this suggested that these tumor models, which were really quite similar, both generated from stem cells, both generated by infecting those stem cells with MYC and another oncogene and transplanting them, that even though these models were made in a similar manner, they were really quite different with respect to the immune response. And we proved this because we took the MG model, which was normally rejected in an immunocompetent mouse, and we knocked out the T cells in that mouse and then showed that the tumors could grow, and all the mice now died of the disease. So this suggested that the MG tumors were not growing because they were being rejected by T cells. So either the MG tumors somehow attract an immune response, or the MP tumors somehow suppress an immune response that would otherwise be taking place. So to figure this out, we did what we call molecular swapping experiments. We knew that the MYC-P53 tumors and the MYC-GFI1 tumors differed only by one molecule, either P for P53 or G for GFI1. So we took the GFI1 molecule and we put it into the MP tumors and asked, does that promote tumor rejection? At the same time, we took the P53 molecule and put it into the MG tumors and asked if that allowed them to grow. And here are the results of those experiments. Here are the MP tumors growing in immunodeficient and immunocompetent mice. They grow in both. And if we put in GFI1 and ask, does this now make the tumors be rejected? The answer was no. The tumors grow with exactly the same latency and exactly the same penetrance, regardless of whether they have GFI1. In contrast, when we put in the P53, the mutant P53 molecule into the MG tumors, what we found is that in the normal MG mouse, the tumors don't grow in a mouse with an immune system. But if we put in this P53 mutant gene, now the tumors do grow. So this was a surprise, but also a revelation, because it said to us that somehow this mutant P53 molecule was making tumors invisible to the immune system. It was suppressing the immune response. And the question was, how? So we entertained a bunch of different hypotheses, and we tested a bunch of different molecules that might be involved. We thought maybe the P53 was inducing production of immunosuppressive cytokines. It was turning on molecules on the surface of tumor cells that suppress T cells, or it was shutting down molecules that are necessary to stimulate T cells. And we looked at a whole bunch of molecules that have been previously demonstrated to play a role in either T cell suppression or T cell activation. And I won't go through these in detail because the bottom line was there were really no differences between MP and MG tumors in expression of any of these molecules. And so we were scratching our heads until we decided to do really the simplest experiment possible. And this is the simplest experiment if you've ever taken a course in immunology, but if you haven't, here's what you need to know. All cells in your body, including tumor cells and not including red blood cells, express class 1 MHC molecules on the surface of the cell. Class 1 MHC molecules' job is to present peptides from inside the cell on the surface of the cell so that the immune system can go around and check if the cell is normal. And so when a cell expresses MHC class 1, if a T cell comes along and recognizes it, it will kill it. But if you have a cell that has no MHC class 1 on it, the T cell cannot see that cell. The T cell receptor has nothing to recognize, and the cell will escape immune surveillance by T cells. And so the bottom line is tumor cells that lack MHC class 1 are invisible to T cells. And what we found when we compared MHC class 1 expression on our two tumor models was a big difference. The MP tumors, which are able to grow without being rejected, had no MHC class 1 on their surface. The MG tumors had perfectly normal levels of MHC on their surface. And if we put this mutant P53 into the MG tumors, it drove down expression of MHC class 1. And these are, if you're not familiar with them, FACS plots or flow cytometry plots that show histograms of the level of expression of MHC. This was not only true in mouse cells, and it was not only true with mutant P53. We took some of our human medulloblastoma cell lines, which had MHC class 1. We put in an shRNA to knock down expression of P53, and with 2 different shRNAs, we reduced expression dramatically, suggesting that it was really the loss of P53 that was resulting in loss of MHC class 1 on the surface of these cells. Not only does this work if you do it in the lab, but if we just take patient-derived xenografts or patient tumors straight from the operating room, and we look at MHC expression, if those tumors have normal P53, they have varying levels of MHC class 1. But if they have mutant P53, they have no MHC class 1 on their surface. And so this suggested to us that P53 was somehow tightly regulating expression of MHC on the surface of cells. Then we started to look at the mechanisms of this and had another surprise. We found that when we compared the mRNA that encodes MHC in MP and MG tumors, there was really no difference in mRNA expression. Moreover, when we looked at the protein levels within the cell, there was no difference in the levels of MHC protein between MP and MG tumors. So this suggested to us that there was some other kind of regulation going on, some kind of post-translational regulation. And so without going into too much detail here, I'll just describe the pathway by which MHC gets to the cell surface. And so within a cell, there's a machine called the proteasome that chops proteins up into tiny little peptides. Those peptides get transported into the endoplasmic reticulum, the ER. In the ER, they get further clipped by an aminopeptidase that makes them fit into the pocket of the MHC molecule and allows the MHC molecule to get to the cell surface. And it turns out that two of the important components of this pathway, the transporter TAP1 and the aminopeptidase ARAP1, are targets of P53. And so knowing this, it then became unsurprising that in a cell with P53 mutations, there was no MHC on the cell surface. And so we proved this in our cell system. We showed that MP tumors have very low or no expression of ARAP1 and TAP1. MG tumors have plenty of those. This is the mRNA level. This is the protein level. And again, MP has little, MG has a lot. And when we do that trick where we put in mutant P53, we drive down expression of ARAP1 and TAP1 at the mRNA and protein level. ARAP1 is particularly critical for expression of MHC on medulloblastoma cells because if we overexpress ARAP1 in those MP tumor cells which have no MHC, that allows them to express MHC. And if we take the MG tumor cells which have MHC and we knock down ARAP1, that's sufficient to drive down expression of MHC. This is also true for human medulloblastomas with the help from Michael Taylor's lab. We looked at expression of ARAP1 and TAP1 in human medulloblastoma patients with mutant or wild type P53, and you can see that the patients with wild type have higher levels of ARAP1 than the mutants, the same for TAP1. And in fact, we've begun to look in other cancer types, and this is just analysis of some data from TCGA showing that in myeloid leukemia, head and neck cancer, lung cancer, and stomach cancer, we see the same pattern where ARAP1 is lower in P53 mutant tumors than in P53 wild type tumors. We're far from knowing whether the findings that we have that I've described already and the ones I'm going to describe are applicable in other tumors. We've really focused most of our attention on pediatric brain tumors. But this is provocative because it suggests that the mechanisms that we're defining might have implications for many different types of cancers in both kids and adults. So what I've told you so far is that tumors with P53 mutations have decreased expression of class 1 MHC. Tumors lacking class 1 MHC are invisible to CD8 positive T cells. And that means these tumors might be resistant to immunotherapy. If a T cell can't recognize the tumor, no matter how much you activate that T cell, it still won't be able to kill the tumor. So the question is there anything we can do to overcome this? I mean, one strategy is just to say, well, let's look at all the patients who are coming in to the clinic for an immunotherapy trial, let's look at their P53 status, let's look at their MHC expression, and if they don't have MHC, let's just send them home. But what we'd really like to do is find a way to rescue this, find a way to overcome the invisibility of these tumors, make them visible to the immune system so that they can be attacked. And so we did a lot of studies to try to find things that might increase the expression of MHC on P53 mutant tumor cells. So shown here is MHC class 1 expression on the two models I've been discussing. Here are the MG tumor cells, which have MHC, and the MP tumor cells, which have mutant P53 and have no MHC. And we found, I won't go through all the reagents that we've tested, but we found that when we treated with one particular agent, tumor necrosis factor, or TNF, we could increase the expression of MHC on the cells that already have it, but most importantly, we could rescue expression of MHC on the cells that didn't. So this was striking because it gave us an opportunity to now make cells that were not going to be recognized by the immune system recognized and rejected. So how does this work? How does TNF induce MHC on a tumor cell? And I'll just go through some of the mechanistic studies we've done, the first of which may not be surprising, and that is if the cell is not expressing MHC because it has no ARAP1 or TAP1, it would make sense for TNF to work by rescuing expression of ARAP1 and TAP1. And that's what it does. Again, this is shown at the mRNA level. TNF-treated cells have much higher levels of ARAP1 and TAP1. And this is at the protein level. And again, a significant upregulation of these proteins, which allows peptides to get loaded into MHC and MHC to traffic to the cell surface and present peptides to the immune system. Now, TNF has been studied for many, many years. It's known that it can bind to at least two distinct receptors called TNF receptor 1 and TNF receptor 2. Each of these has a different machinery attached to it and a different signaling pathway downstream of it. And so we wondered which of these receptors is important for mediating the effects of TNF on MHC expression. And we found that if we took TNF, of course, it can bind to both of these receptors and it can induce MHC expression. But if we took a ligand that could only bind to TNF receptor 1, it had no effect. Whereas a ligand that could only bind to TNF receptor 2 was as potent as TNF at inducing this response. So this suggested that the key receptor was TNFR2. And that's important because TNF has many, many different effects on almost every cell in the body. But most of the cell death effects and most of the toxicity that's seen with TNF appears to be mediated by TNFR1. So if we were looking for a more specific way to trigger this response, the fact that we could activate TNFR2 might be a way to get a cleaner response without some of the side effects. And I'll talk more about that in a few minutes. We also knew that TNF can function intracellularly by activating at least two, and in fact, many more than two, signaling pathways downstream to induce gene expression. And people refer to the two major pathways as canonical and non-canonical. Obviously, this one was discovered first. The biggest difference is that the canonical pathway results in translocation of this dimer of P50 and REL-A into the nucleus where it can turn on target genes, whereas the non-canonical pathway turns on P52 REL-B through a different mechanism. In previous studies, it's been shown that TNF receptor 1 is the main activator of the canonical pathway. TNFR2 can also activate this pathway. I guess I lost that other piece, but whereas the non-canonical pathway is typically only activated by TNFR2. So we were sort of guessing that this was going to be the important pathway downstream of TNF. And it turns out that we were wrong. So if we looked by chromatin immunoprecipitation at what was bound to the ARAP1 and TAP1 promoters following TNF treatment, we found that TNF induced a very significant binding of REL-A and P50 and not REL-B and P52 in the nucleus. So this suggested that REL-A and P50 were the key mediators. And we added further evidence to this by knocking down using shRNA, REL-A or P50. And we showed that each of those could dramatically reduce the induction of MHC by TNF, whereas knocking down REL-B had no effect. Having done our mechanistic studies on mouse cells, we wanted to make sure that the effects that we saw of TNF on MHC were also true in human cells. And so these are two of our p53 mutant patient-derived xenografts. And you can see that when we treat them with TNF, we see a marked upregulation of MHC expression. And this is a patient sample. We've begun to capture samples from the operating room, bring them back to the lab, and immediately use them in experiments. And in this particular patient who also has a p53 mutation, treatment with TNF results in a very significant upregulation of MHC expression. So this is not only true for our mouse models, it's also true for human medulloblastoma cells. And interestingly, we've begun to look in collaboration with Oren Becker's lab at Northwestern and with really generous support from Alex's Lemonade Stand Foundation, we've begun to look at brainstem glioma and supratentorial high-grade glioma. And this just shows in a p53 mutant mouse model that Oren's lab developed, when we treat with TNF, we upregulate MHC expression. And in a p53 mutant human DIPG line, again, we see a significant upregulation. So this ability of TNF to rescue MHC expression may be a phenomenon not just of medulloblastoma, but of other tumors as well. So if TNF can rescue MHC expression on tumor cells, can we use this strategy to enhance responses to immunotherapy? Now, those of you who've been around for a few years may have heard about TNF and its effects on people. TNF induces what we now like to refer to as cytokine storm. That is, it causes massive system-wide inflammation that can, if uncontrolled, lead to death of a patient. And this was the reason that when TNF was in trials about 25 years ago, it was rapidly withdrawn from the market because it seemed to not do what its name advertised, that is to necrose or kill tumors, but it did seem to cause very significant side effects in the patients who received it. And this was true, there were trials in both adults and in children with cancer. And unless you used a very low dose, you saw these severe side effects. And if you used a very low dose, then you saw no effect of TNF on tumor growth. So why are we even considering adding this to immunotherapy, which on its own can cause a cytokine storm? And the reason is that the doses that we need to induce MHC on a tumor cell are at least 50 to 200-fold lower than the doses that have ever been shown to cause toxicity in a mouse or in a patient. And that means that we should be able to at least safely expose patients to these doses of TNF and see whether it can enhance immune responses. And so we tested this first, of course, in our mouse models. And this just shows that if we take a mouse with a tumor and we inject low doses of TNF into that mouse, this is 500 nanograms per kilogram, which is about 2,000-fold lower than the dose that causes toxicity in a mouse. Wait 24 hours and then look at MHC expression, we can induce MHC in this tumor-bearing mouse. And that's really important for two reasons. One, because the mouse had no side effects from that injection. And two, because the TNF was administered systemically into the circulation, and it was able to get into the tumor in the brain and cause upregulation of MHC, which means that TNF can cross the blood-brain barrier. And in fact, there's a significant literature that says that TNF can cross the blood-brain barrier, but we wanted to validate that in our own lab. And so if TNF can induce expression in vivo, the question is, can it be used to enhance responses to immunotherapy? And so this was the, as they call them, money experiment, where we took mice with tumors in their brains and we treated them with either nothing, with the immune checkpoint inhibitor anti-PD-1, which is a really good way of activating T cells so that they're more effective at killing their targets. And you can see that anti-PD-1 really does very little to tumor growth in these animals. And that's because no matter how active the T cells are, if the tumors are invisible, the T cells can't kill them, so the tumor keeps growing. If we treat with TNF, we see some shrinkage of the tumor. We think that's because the TNF is exposing MHC on the surface of the tumor and allowing the immune cells that are there to attack them. But the really significant effect came when we treated with TNF and anti-PD-1. That is, we gave a shot of TNF And then the next day we gave anti-PD-1. And we repeated this every 3 days for at least 4 weeks or as long as the animal survived. And so what we found is that in this scenario, and these are mice 3 weeks after the beginning of treatment, the tumors are essentially gone. And if we wait even longer, you can see that in mice that receive the combination, the tumors never come back. In mice that receive TNF alone, there's prolonged survival, but eventually they outpace the immune system and keep growing. But this combination therapy is able to eradicate tumors in our model. This was true for medulloblastoma model. It seems to be true to some extent for Oren Becker's brainstem glioma model as well. So this is induction of MHC in an intracranial transplant of Oren's mouse model of DIPG. And then here's that same experiment that I just showed you for medullo. And you can see control animals, anti-PD-1 does nothing, TNF does a little, the combination does a lot. These animals are tumor-free by 3 weeks. About half of them end up coming down with tumors, but half of them are long-term tumor-free. And for DIPG, a 50% cure rate would be unheard of. So just to summarize what I've told you, and I can't see the clock, so I have no idea where we are in time, but just to summarize what I've told you, we found that P53 regulates expression of these 2 key components of the antigen processing machinery, ARAP1 and TAP1, and thereby determines whether a tumor cell will express class 1 MHC on its surface. P53 mutant tumors lack these proteins, so they lack MHC, and they're able to grow in immunocompetent mice. P53 wild type tumors get rejected in those animals. The lack of functional P53 may render tumors invisible to T cells and insensitive to immunotherapy. And TNF can rescue expression of MHC on P53 mutant tumors and enable rejection by the immune system. So we've done all of our studies in mice, as we should when we're starting. The question is, can this have an impact on patients in the clinic? And so we've been in discussions and working really hard with the Pacific Pediatric Neuro-Oncology Consortium, or PNOC, over the last couple of years to design a trial to do this in kids with medulloblastoma. And the design is like this. We take a biopsy from the patient's tumor. This is at recurrence. We take a biopsy from the patient's tumor and look at expression of MHC. We also assay for immune infiltrate and P53 mutation. Then we give 1 shot of TNF, and we look through another biopsy or through a resection at the levels of MHC to see if they went up. And then we give the cycles of TNF and anti-PD-1 that we gave to our mice, and we see whether that results in tumor eradication. The hope is that in a patient, TNF will get into the brain, it will drive up expression of MHC, and it will render cells sensitive to T cells, which can be activated by anti-PD-1. The trial is in, I would call it, the late stages of planning. We're going through various regulatory steps to get approval. We have approval to get the therapeutic agents, the TNF and the anti-PD-1, from companies that make them. And we're really excited to see whether this might benefit kids with recurrent medulloblastoma and ultimately kids with other brain tumors. So that's all I've got. I want to thank the people who have done this work. Yan-Sin Pei was a fantastic postdoc in my lab. She's now a faculty member at Children's National Medical Center in Washington. She did all the work developing the MIC-P53 or MP model. Catherine Lee was a grad student and then a postdoc in my lab. She designed and developed the GFI1, the MG model, and is now a scientist at Pfizer. And Alex Garanche is a postdoc still in my lab, but interviewing for jobs as we speak. And she did basically all the work I showed you today, using the 2 models and a bunch of PDX models to look at immune responses to medulloblastoma. I also want to acknowledge the other members of my lab who, when I tell them to jump, they say how high. And all of our collaborators who enabled this work, and the fantastic funding sources, and in particular Alex's Lemonade Stand Foundation for their untiring support of our work. And thank you all for your attention. I'm more than happy to take questions.
Thank you, Rob. That was an excellent talk. Very exciting work. Hoping that it shows more promise as the clinical trials get started. And I just closed my chat window and I lost the questions. Hold on. Okay, here we go. Got a bunch of questions coming in. So
Okay.
we have, I think we have about 20 minutes. So
Yep.
We can get to a bunch. Okay, the first question that came in early was from Ben Myers, and it says, have you looked at MHC class 2 in these tumors?
Yeah, that's a great question. Let me see if I can, I can't see anybody or anything right now. So let me see whether, or is there anybody to see?
There's no one to see. These are all coming in through the chat.
Okay, got it. Well, no, okay, now I can at least see you, Jay, so I can.
Okay, I see what you mean. Yeah, I messed up my screen also. There.
Yes, so we have looked at MHC class 2, and there's a short answer to that and a much longer, more interesting answer that I probably don't have time to go into. But the bottom line is these tumors, like almost all tumors in the body, or most cells in the body express class 1 and not class 2. There are a small number of immune cells, in particular dendritic cells and B cells, that express class 2 MHC and use it to present antigen. But it is true that in some tumor types, MHC class 2 is expressed or can be induced. And in those tumors, there can be recognition and even killing by CD4 T cells, which like to see antigens in the context of class 2. We have looked at our tumors and they do not express class 2 MHC. We can induce it if we treat them with significant doses of interferon gamma for a few days. And so it's conceivable that tumors might express class 2, but at least at baseline level, they do not. I'll sort of tease the bigger story, but I really can't go into it. And that is that we have found, in addition to an important role for CD8 T cells, we found a really important role for CD4 T cells in tumor rejection. We don't think that that's through direct recognition of tumors by class 2, via class 2. We think that it's through an antigen presenting cell. And we have a little more detail, but we don't fully understand it yet. So thank you for that question. That's really interesting.
Sorry, I couldn't get unmuted there. The next question says, amazing talk. I have a question concerning neoantigens. One challenge in pediatric tumors is that we barely have mutations. Did you check what is presented on the MHC molecules? Could it be that the tumor cells end up presenting self-antigens and therefore are still invisible from the immune system?
That's a great question. So we talk a lot and people like to show sort of tumor mutational burden and suggest that tumors that have low mutational burden are at least less, if not unsusceptible to T cell attack. It's certainly true that if there are a lot of neoantigens, a lot of mutant proteins, the immune system has a lot more to choose from. But a lot of studies over the last 30 years of immunology have suggested that you don't really need a lot of antigens, you just need one or two good ones. And the other thing is that the immune system does get tolerized to a lot of self-antigens, but it doesn't get tolerized to all. It tends to recognize developmental antigens that are silenced early in life and get aberrantly turned on later in life. It can recognize those as well. And so the notion that pediatric cancers and pediatric brain tumors are not susceptible to the immune system, I don't think is quite correct. That said, I do think that we need to do a lot of work to try to understand what are the antigens that are overexpressed or aberrantly expressed by tumor cells so that we can enhance immune responses to those through either vaccination or some other mechanism that drives up immune responses to tumor antigens. And so we've begun to do a lot of work. We don't yet know what is the antigen that's being recognized in our mouse model, and we'd love to know, so we're trying very hard to identify it. But we're also doing similar studies in our panel of patient-derived xenografts to see if we can identify commonly presented peptides in human medulloblastoma cells that might be targets for some kind of vaccination. I think that ultimately, to get an effective immune response, we're not going to be able to take one approach. We're not going to, checkpoint inhibitors alone are not going to work for most patients, in part because they may not present antigens. Even driving up MHC expression may not be sufficient if there aren't good antigens for the MHC molecules to present. And so then how do we drive up the effective immunogen concentration in a tumor? And I think you can imagine we could do this by causing more mutations, say through radiation, by bringing in foreign antigens like with an oncolytic virus, or by vaccinating to rev up responses to antigens that may not have gotten the attention of the immune system. And so I think that end of the game, the finding the right antigen and driving an immune response against it is still a work in progress. And until we make some progress on that, I think we're still gonna suffer from incompletely effective immunotherapy. I hope that answered your question.
Thank you. The next question came from Anirban Das, and it says, thank you for the great talk. Does the combination of low-dose TNF and PD-1 inhibition increase the risk of systemic cytokine storm?
That's a really good question. It's a question that's in the forefront of our minds in the context of the clinical trial. Cytokine storm usually results in very severe, very obvious side effects very quickly after you give TNF. So what I can say is with these doses of TNF in our mice, we see no side effects, and we've done daily treatment for 30 or more days. And we don't see any sign that the mouse is perturbed by this. Those are mice, and people are people. And so we're very, very sensitive to the possibility that there might be side effects in patients. So this is part of the reason for the trial design that I went through quickly, which is that we'll give a single shot of TNF on its own first. That will allow us to see if TNF affects MHC expression, but it will also allow us to see if on its own, these doses of TNF have any side effects for the patient. And if they don't, which we expect based on the previous clinical trials, that will at least allow us to conclude that on its own, this dose of TNF is safe. Now, whether the combination of TNF and anti-PD-1 leads to a more severe cytokine storm, I think that's a really important question. And unfortunately, the only way to answer that is in the context of a clinical trial where patients are given these therapies and really closely monitored. The safety of the current dosing regimen of PD-1 in kids with medulloblastoma and other pediatric brain tumors, the safety record has been quite good. There have not been very severe side effects in those kids, but there has also been no efficacy. And so this is, I think it's a critical question, and it's obviously the primary goal of the trial. We'd love to see this combination cure patients, but the primary goal of the trial right now is safety. Can we give these doses safely and not cause any side effects for these kids?
Okay, so the next three questions are surrounding P53. First one from Christopher Park. Do you have thoughts that this would have benefits outside of the P53 aberrant populations, such as other subgroups of medulloblastoma, group 3?
Yeah, that's a really good question. It's something we've investigated a little bit. And I see the next question, I might touch on that as well. But we do think that this may have benefit outside of P53 mutant patients for two reasons. One, because even patients who lack actual P53 mutations often have mutations in other components of the pathway that may lead to a P53 mutant-like phenotype. And second, even in our model, which has wild-type P53 and has MHC, we see a significant increase in expression of MHC when we treat with TNF. Now, whether going from no MHC to MHC presumably can have a significant effect on the ability to be recognized by the immune system. Going from some MHC to more MHC, we really don't know if that's beneficial, but you could imagine that it might be. Because you could imagine that if there is a neoantigen or an important antigen that is not very abundant in the cell, it may not be able to compete effectively to get presented on the surface if there's only a little bit of MHC. But if you cause a 10-fold increase in the MHC expression in those cells, now you may be able to present less abundant antigens, including key neoantigens or tumor antigens that the immune system needs to recognize. So again, we don't know. We'll learn that a little bit from the trial, because the trial does not rule in or out P53 mutant patients. So those patients we will have patients that have both wild type and mutant P53, and we'll be able to see whether that upregulation happens. It certainly does happen in the cells that we've treated in culture. Let me tackle Jerry Shea's question, which is knockout of P53 the same as mutant P53? And the answer is, so a lot of our studies were actually done not on knockout, but on dominant negative P53 expressing mouse tumor cells. And in that context, we're actually driving expression of something that very potently suppresses endogenous P53. However, we do see the same effect on MHC expression with P53 shRNA, so knocked down. And importantly, we see little or no expression of MHC in P53 mutant patient-derived xenografts and patient samples. And in that case, of course, it's naturally occurring P53 mutations. Now, we've begun to probe that more deeply, and we don't have definitive answers, but there are P53 mutations that, not all P53 mutations are the same. And there are P53 mutations that have more or less potent effects on MHC, on ARAP and TAP and MHC expression. So I think as we go further into this, we're going to need to learn which P53 mutations are actually most potent at driving this and which P53 mutations are not. So hopefully that answers that question as well.
Thanks, Rob. So did you answer the next one also in your previous answer? Where do you think P53 wild type tumors in humans downregulate MHC?
No, I don't think I did.
Okay.
And it's a really good and actually really big question. So I think that downregulation of MHC is a major mechanism by which tumors evade the immune system. And P53 mutations are only one mechanism by which that can happen. There are certainly tumors in which the MHC molecule or its partner beta-2 microglobulin are mutated, and therefore the MHC cannot be expressed. And in those tumors, TNF's not going to make any difference. There are also other elements of the antigen processing machinery besides ARAP1 and TAP1, and there are tumors in which those are mutated. And again, in those cases, we're going to need to find alternative mechanisms to re-express MHC. One of the things that's been published recently after analysis of large trials of anti-PD-1 treated patients is that those patients who become resistant often become resistant because they reduce expression of MHC1 on the surface. So tumor cells know that this is a really good way to hide themselves. We just need to figure out how they're hiding themselves, and P53 mutation and ARAP and TAP are one strategy. But in tumors where that's not happening, where MHC is silenced through some other mechanism, we're going to need other mechanisms to either get MHC expressed or get the immune system to recognize the tumor cells in an MHC1 independent manner. And that could include CAR T cells that don't use MHC, that could include natural killer cells, or that could include therapies that activate myeloid cells like CD47, for example.
Okay, the next question says, nice talk, Rob. A couple of questions. Are there any other cell surface molecules that are downregulated in MP tumor cells? ERAP1 and TAP1 may mediate protein transportations for many surface proteins, and it looks like the MHC1 inducing effect of TNF alpha is P53 dependent. What happens if you treat MP plus B6 mice?
Okay, so it is true that ARAP1 and TAP1 clip and transport other peptides or peptides for other purposes besides loading them into MHC. We haven't studied those in a lot of detail. There was an interesting paper recently showing that ARAP1 plays an important role in sonic hedgehog signaling. And so in those tumors, the role of ARAP1 in activating the hedgehog pathway is going to be really important to pay attention to. But we haven't looked extensively at other proteins that whose expression on the surface may be affected by ARAP1 and TAP1. The second part of the question, excuse me, looks like the MHC1 inducing effect of TNF alpha is P53 independent, and that's true. So if we look at the promoters of ARAP1 and TAP1 in a P53 wild type cell, they're occupied by P53 and that turns on expression. In a P53 mutant cell, they're not occupied and they don't express those genes. And then when we treat with TNF, the promoter gets occupied by NF-kappa B, by REL-A and P50, and that turns on expression. So there are different signals going into the promoters of these genes, depending on the circumstances. I'm not sure I understand what happens if you treat MP plus B6 mice. So maybe you can email me afterwards and we can touch base about what you were asking there.
Okay, going back to the cytokine storm, I think they've had similar experiences with the CAR T cell treatments. And so could you manage them the same way, do you think? That's coming from me, by the way.
Yeah, so yes, I mean, cytokine storm is a prevalent problem in immunotherapy. It's a very serious problem, but if you're aware of it, there are ways to try to manage it. Now, it's easy to say that, and in some patients it can't be managed, but we have antibodies against some of the cytokines that mediate the storm, that mediate the systemic inflammation. And so if you have those at hand or you give those to the patient in advance, you can actually block the systemic side effects without necessarily affecting the effects of the drug. But it is, I mean, I think it's to me testament to the fact that although it's convenient to give these therapies systemically, we really ought to be looking at ways to give them locally. And we're looking at that as well. But I think that if there were a way to deliver the CAR T cells, the TNF or whatever, into the tumor bed itself, or at least in the vicinity of the tumor, we would worry a lot less about the systemic side effects, because the concentrations that would then leak out from the tumor to the rest of the body are, would be lower.
Okay. Next question is, why is PD-1 targeted instead of PD-L1?
So we don't, I mean, there's not a good reason. We tried PD-1 and it worked. PD-L1 binds to PD-1, as I'm sure you know, and you can block either partner. PD-1 is on the T cells. PD-L1 could be on the tumor cells, but it could also be on other cells in the microenvironment. And so we are in the process of testing whether PD-L1 does the same thing in our system as PD-1. It may or may not, but it's a really interesting question.
Next question asked if the lecture recording will be available, and I think it is going to be available. Is that right?
Should be.
Yeah, okay. Last question is from Rob Castellino. It says, great presentation, Rob. What's the MHC1 expression in Leaf-Romani patients? Are they more susceptible to toxicity from your approach? And would you need to screen for germline P53 alterations in your trial? Yeah,
so that's a really good question, Craig. The MHC, we haven't looked extensively at MHC expression in other parts of the body in Leaf-Romani patients. We have looked in P53 knockout mice, and in some tissues there is no MHC, and in other tissues there is MHC. So there's some complexity there. In Leaf-Romani patients, typically they are, throughout the body, they're P53 heterozygous, so they still have P53. And then in the tumor, they lose the other copy. And one of the tumors that they get when they lose the other copy is medulloblastoma, typically hedgehog driven medulloblastoma. So among the PDXs and the patient samples that we've looked at, some of which I showed today, some of those are leaf Ramani patients, and we do see this upregulation. Whether they would be more susceptible to toxicity, I'm not sure, because I think the toxicity, the systemic toxicity, would be affecting cells throughout the rest of the body that are likely to be p53 heterozygous. It would have the maximal effect, at least in the sense that we've been looking, on cells that are p53 mutant, which would be tumor cells. We will certainly look for germline p53 alterations as well as somatic alterations in our trial, because we want to know whether those patients are more or less susceptible to the efficacy as well as the toxicity.
Well, that brings us to the end of our questions. And it brings us to the end of our hour. So it was perfect timing. Thank you again, Rob. We appreciate it. It's a great lecture, and thank you for everybody who joined us. And hope everybody stays well and stays safe.
Thank you, Jay. It's been a pleasure.
Thank you.